My name is Quinn O'Connor. I am a junior at Sarasota High School, and this is my submission for the 2021 Science Fair, in which I will be comparing the effectiveness of a 3D printed versus an industrial Agarose electrophoresis chamber. This is a technique that is most commonly used by forensic scientists and biologists in order to study the pattern of DNA macromolecules as they migrate across an electromagnetic field. The electromagnetic field causes the DNA molecules to separate by size as they go from negative to positive charges. Electrophoresis is more commonly used when analyzing DNA that is found at a crime scene, but can also be used in studying the relationship between proteins and antibodies, in testing vaccines, and in looking at hereditary disorders. The purpose of my project was to find a more cost-efficient way to analyze DNA with cheaper and more effective equipment so that we can spend less money on buying the actual machinery and more money on research and labs. My engineering goals for my project were to 3D print an equally effective electrophoresis chamber that had the same efficacy as the industrially manufactured counterpart, and to make the 3D printing project a lot cheaper than the purchasing of an actual chamber. Electrophoresis chambers can range up to thousands and thousands of dollars, but if we can minimize costs in our machinery, we can implicate that money into our actual research. My independent variable was the type of chamber and the amount of magnetic charge. I did have to change this a few times to see what worked best. My dependent is the consistency between the macromolecule change and how far they migrate. My control was the buffer solution and the Agarose technique. I was very careful about how much of each I was using, and the constant is the amount of buffer solution and time per trial. So first for my methods, I had to create the Agarose gel after I had set up the chambers. For the industrial chamber, I combined 1 gram of Agarose with 100 milliliters of buffer solution, and for the 3D chamber, I made that half by putting half of a gram of Agarose solution and 50 milliliters of the buffer solution due to the size. They both went into the microwave for about 1 minute. I measured the amount of bubbles that were coming up to know that the chemical reaction was occurring, and then I let them cool. It takes anywhere from 5 to 35 minutes. As the gel is hardening, you must place the well comb into the gel so that it will stand upright and create small wells for your food coloring to go in. The food coloring will act as the macromolecules in our experiment, as we did not use real DNA. Fill each chamber with buffer solution so that the gel is completely covered. The amount of buffer solution will differ based on the size of your electrophoresis chamber. I used 75 milliliters for my 3D printed and about 800 milliliters for my industrial. In order to run the 3D printed chamber, you must cut 2 pieces of stainless steel wire so that they are longer than the width of the chamber itself, and bend the 2 wires so that they can go into the small holes on the side and hit the buffer solution. 1 wire will act as the negative electrode and the other will act as the positive electrode. Now connect the battery pack, and they should be connected in a motion as seen here. And place 1 alligator clip on the negative terminal, and then the other side of the alligator clip onto the negative terminal of the battery, and do the same for the positive. For the industrial, all trials should be conducted at 125 volts of electricity, and you can follow the instructions for the setup as seen in the user manual. To collect data, I checked the gel's progress after 40 minutes, and I measured the amount of bands produced as well as the amount of migration in millimeters. I recorded all of my observations in my logbook, and then I would have liked to repeat more trials. I would replace the 5 batteries after every new trial to ensure that the electric magnetic charge is fresh. So after conducting 4 trials per each chamber, this is my data. The number of bands did increase per each trial, as well as the distance migrated in millimeters. It should be noted that the industrial chamber did produce more bands, except for in trial 3, but they were still very close, and the distance migrated did increase per each of them, but in the industrial chamber, the distance migrated was higher. So these are my graphs. On the left, we have total distance migrated in millimeters, and on the right, we have number of molecules migrated per each trial. As you can see, in my first trial, the 3D printed didn't even work we had battery issues, so I had to get a whole new battery pack and retry that. But I kept the data in here for the purpose of the experiment. Trial 4 was by far my most efficient trial, where I had the most bands produced per each chamber, as well as the furthest migration. Some limiting factors could have been the human error in my experiment, which I will get to later, as well as the industrial chamber is a lot larger than my chamber, and so it had more room to migrate in the first place, which is why I never could have reached the same results with my 3D printed chamber. So these are my results. Contrary to my hypothesis, the industrial chamber did produce more accurate results more quickly. In trial 1, the industrial chamber macromolecules moved 7.3 centimeters, and the 3D chamber wasn't even run due to human error in the agar concentration and the batteries. In trial 2, the industrially manufactured chamber molecules moved 5 millimeters, where the 3D printed chamber moved 7. So this was kind of an outlier in my experiment, but it was likely due to some malfunction with the industrial chamber battery. In trial 3, the industrial moved 6 millimeters and the 3D moved 2.1 centimeters. And in trial 4, the industrial chamber moved 7.9 centimeters, whereas in the 3D chamber, it only moved to 2.9 centimeters. However, this was my most effective trial. As you can see, the numbers did proportionally increase. At higher voltages, the 3D chamber produced longer migration rates, but it was still not as efficient and precise as the industrial chamber. Also, the agar and buffer ratio drastically affects the band visibility, so I would like to play around more with different concentrations of agar to see what gives me the best results. I had minimal error in my experiment, and as you can see on the top, we have my 3D printed trials, and on the bottom, we have the industrial trials. You can see that there was a lot more room for migration in the industrial chamber, as well as you can more distinctly see the bands. So my biggest error was that I discovered a wiring issue on the positive charge side of the industrial chamber, which may have caused some irregularities in the data from the second and third trial. I also had to reuse some buffer solution due to the underestimation of how much I would need for my experiment, and this could have caused some altercations in my data as well, because it could have made the electromagnetic field weaker. In future applications, I hope to implicate viable biological material within the experiment and to make a chamber that is the same scale as that of the industrially manufactured machine. I would conduct more trials with varying agar/buffer ratios, which I did discuss earlier, to see what concentration of agar in the gel has the clearest results. I would also make modifications to the 3D printing model that I used, which I did find it is cited in my citations, and I have that template on the next slide. I would like to increase the size to include 2 testing sites. As the industrially manufactured chamber, you can stack 2 plates of agar gel to test 2 DNA molecules at the same time. I would increase the cone size because I want to improve handling, and I think it would be more manageable as it was slightly too small. And I also would add some sort of suction for the wires to stay in place, as they were extremely tedious. In conclusion, the 3D printed chamber was not as efficient as the industrially manufactured chamber, but this was likely due to human error as well as the lack of the electromagnetic charge as a result of the smaller battery power. I had a total of 75 volts for my 3D printed chamber, because that is the amount that I could get with the batteries all packed together, whereas in the industrial chamber I used 125 volts. However, with more experience and more experimentation, I think that I could use the voltage variation and different tracking dies in order to prove that my chamber could work just as well. It was relatively successful, and all of my printing filament did withstand the electromagnetic charge, as well as did not have any leakages with the buffer solution. Here are the modifications that I would make. I would double the size of the chamber so that I can run 2 tests at once, and I would also improve the size of the comb so that my well sizes would be bigger, as well as the width of the comb. And these are my references. Thank you.